start with the session. Mamuna, was you there in last class, or you missed it?
Sir, I was there.
Yeah, he was there. Fine. So today we were supposed to start with the chloride shift. Yes.
Yes.
Fine. Right. So let's start it. Fine. So let's give a quick recap of the transportation of the carbon dioxide. Actually, transportation of the carbon dioxide is the most important topic of this one. And for your medical examination, they are more interested in. I'll give that kind of the question, previous year question, even into previous year questions which have been asked already. So we'll discuss some questions as well. Right? So one more. And I think we have discussed already the assignments which you submitted, you people submitted, yes or not? Yes, guys.
No, sir.
We haven't discussed that. Fine. So let's start today's session. Then let's finish this chapter. We are at the end of the chapter, and after this we have to do some of the disorders, and then definitely I'll open your assignment as well. Then we'll start with the circulatory system. Right. Chloride shift. So look, in last class, we have discussed that transportation of the carbon dioxide takes place in three forms, right? So in transportation of the carbon dioxide, let me make it larger. Look, in last class, we have discussed that transportation of the carbon dioxide takes place in three forms. First, the 7% of the carbon dioxide, because solubility of the carbon dioxide is more than the solubility of the carbon dioxide is more than the solubility of oxygen, and we have noted down that thing as well. So carbon dioxide is more soluble with the blood plasma. In fact, the water of the blood plasma. Then 7% of the carbon dioxide is transported from the tissues to the lungs, or we can say alveoli, with the help of in dissolved condition. Rest 70% is transported as a bicarbonate. Hence, the contribution of bicarbonate ion have a larger chunk or loin share in the transportation of carbon dioxide. And last but not the least, which we were studying since our ninth class, that 23% of the carbon dioxide is transported with the help of hemoglobin. Previously, in the lower classes, we used to understand that all the carbon dioxide and oxygen are transported by the help of hemoglobin. But this is not the true, as you can see. So, large chunk of the carbon dioxide is transported with the help of bicarbonate. How does the bicarbonate help? Again, so we were discussing in last class how the bicarbonate ions are formed. So the carbon dioxide react with the water which is present in the uh, inside the uh, RBC, that is in the plasma of RBC. So this carbon dioxide react with the water with the help of enzyme called carbonic anhydrase, and this enzyme have been asked in medical examination. So carbonic anhydrase is an enzyme which is already present in our blood, already present in inside the RBC. It changes carbon dioxide into carbonic acid, and this is a reversible reaction. One more important thing, that means it always remains in equilibrium state. Both of the component are found in equilibrium state, right? When the equilibrium will shift, again the reaction will go ahead. Again, the carbonic acid is very unstable compound, you know. Then carbonic acid is very unstable compound, and what will happen? Again, in the presence of carbonic anhydrase, which is working over here as a catalyst, in presence of carbonic anhydrase, this carbonic acid breaks down into bicarbonate ion and hydrogen ion. So I have discussed that the hydrogen ion remains in the plasma. Hence, in the presence of hydrogen ion, the pH value will, what will happen? The pH value will decrease because they will become more acidic, right? So if the carbon dioxide, if hydrogen ion will increase, so as we know that pH value is inversely proportional to the concentration of the hydrogen ion. The hydrogen ion will rise and pH value will decrease. Because of these free hydrogen ion, the pH value of deoxygenated blood becomes from 7.4 to 7.34. It's a slight difference, but comparatively, this is more acidic than the normal blood. Now, that's how the formation of bicarbonate ion takes place. Now, we were about to discuss with the chloride shift phenomenon. How does this phenomena takes place? That's what we are going to study, right? Now, I'll tell you carbon. Put the heading, that the heading is, put a heading, chloride shift. And another name of chloride shift is hamburger phenomenon. And the same question have been asked many times. And the chloride shift is also important for your school examination. They can ask you. Chloride shift, or the same phenomena is known as hamburger's phenomenon. Hamburger's phenomena. Hamburger's phenomenon. Hamburger's phenomenon. So now first I'll explain what is the chloride shift, what is the hamburger phenomenon. For sake of example, suppose that this is our blood cap. So this is the wall of blood cap. So this is the epithelial wall of blood cap. This is epithelial wall of blood cap. Yes, this is the epithelial wall of blood cap. Epithelial wall of blood capillary. Blood capillary. Again, there are tissues. These are the actually tissues. They are the tissues, or you can say the cell of our body. So they are the tissues of cell of body. What will happen? First, carbon dioxide, as we know, between the blood capillary and the cell or tissue, there is a middleman, and we know that middleman is called lymph, right? We will study regarding the middleman more in the circulatory system. I will explain you separately what is the lymph actually is. We have already discussed, and we'll again discuss that thing in elaborate. So the carbon dioxide first diffuses into lymph. So between the cell is not directly contact with the blood capillary, and it do not directly transfer or take anything. Neither it take oxygen directly, nor it gives carbon dioxide directly. It gives indirectly. First, any cell give it to lymph, which is middleman, then it give to capillary. This is the capillary one. They will cross to the capillary. And inside the blood capillary, there is red blood capsule. So RBC is present over there inside the blood capillary. So this is our RBC, right? Now inside the RBC, a phenomena will take place. And what is that phenomena? That phenomena is called enlarged chloride shift. Now we are going to discuss with the chloride shift. What will happen? This carbon dioxide, will go inside the blood capillary. Inside the blood capillary, carbon dioxide, as we have already discussed, in presence of carbonic anhydrase, it will react with H2O, means water, and a reversible reaction will take place. And that reversal reaction, it forms another type of the component, and we know this component is what? This component is H2CO3, means carbonic acid. So formation of H2CO3 or carbonic acid will take place. Again, the carbonic acid will break down into two components. What is these component? That we know this is bicarbonate and S3CO, HCO3, not HCO3. Yes, HCO3 negative carbonate ion, and plus another component that is called hydrogen. This hydrogen, right? First, I want to discuss what will happen with this hydrogen. This particular hydrogen, this hydrogen ion, it will remain inside the cell, inside the carb cell. What will happen this hydrogen ion? This hydrogen ion will combine with the hemoglobin. So now hydrogen ion is going to combine with the hemoglobin, and this is hemoglobin. Along with the hemoglobin, hydrogen ion will form HHB, right? So it will form HHB. So hydrogen ion is going to form HHB, right? HHB is a compound, right? So this hydrogen meet with the hemoglobin and it form HHB. Clear? So what will happen with the rest of the carbonic ion? So this hydrogen, hemoglobin plus H positive, they will form HHB. So HHB compound will be formed inside the. What will happen with this bicarbonate ion? Actually, at the wall of the this RBC, there are certain pump. So these pump, as we know, the function of pump, we have discussed it even in plant physiology, that the function of pump to transport something in and transport something out, or so there may be symport, antiport, we have already discussed in detail, right? What will happen? These pump will push out the extra HCO3, that is bicarbonate ion, outside, and they will bring inside the chloride ion. Please try to understand. What will happen? They will push out of RBC these bicarbonate ion, and in return, they will bring back chloride ions. This phenomena is called chloride shift. Because of the entry of the chloride, water will come inside the cell, means water will start moving inside the cell, and by this way, the cell will get swell. This is called chloride shift phenomenon. Is it clear, or this is called hamburger phenomenon? Is it clear or not?
Yes.
Sir, can you please explain it also?
Again, you want me to explain it again?
Sir, the last part.
Last one. Okay. What will happen? The extra bicarbonate ion, what will happen? The extra bicarbonate ion will be pushed out, right? And in return or in exchange of chloride ion, right? So bicarbonate ion ball will go outside and chloride ion will come inside, right? This phenomena is called chloride shift. Here, the hydrogen will combine with hemoglobin and it will form HHB, right? Due to the entry of chloride ion, now the water will start coming inside. Why? Because the concentration of ion increasing inside means the solution is become more concentrated or hypertonic. Due to this thing, water will gush or water will come inside the cell, and these RBC will swell. They will get turgid. Is it clear? So please do one thing. Please note it down, this one, and let me know. Then I will make you to note it down. And in case if you're a school examiner, they ask this question, then what you have to do before writing down the answer, first you have to draw. This is called schematic diagram. These are not diagram actually. These are called schematic diagram. Schematic diagram are actually those diagram which are not real diagram, but they explain each and every phenomena in written and in flowchart. And they are the blessed explanatory tools of the answer writing. That's why I incorporate all this. I love to incorporate all this. Done.
Done, sir.
Done. Okay. So please start noting now. In chloride set within this diagram, below this diagram, about 70%, about 70% of bicarbonate ion formed in RBC, about 70% of bicarbonate ion formed in RBC enters into enters into blood plasma enters into blood plasma. So out of these red blood capsules, there is blood plasma, right? Blood plasma. And how many? The total bicarbonate ion formed inside, out of them 70% move out, right? Out of the RBC, right? Full stop.
Sir, can you please
Uh, you want me to explain it? Like,
no, sir. Can you please repeat it again?
yeah, sure. About 70% of about 70% of bicarbonate ion formed in RBC, about 70% of bicarbonate ion formed in RBC enters in the blood plasma. Full stop. Excess of excess of bicarbonate ion excess of bicarbonate ion leaves the RBC. Excess of bicarbonate ion leaves the RBC in exchange of chloride ion. In exchange of chloride ion. Full stop. This phenomena is called this phenomena is called chloride shift. This phenomena is called chloride shift. Full stop. Consequently, underline this statement. Consequently, RBC take up water. Consequently, RBC take up water and increases its size. And increases its size. Full stop. That's how bicarbonate ions are transported. That's how bicarbonate ions are transported from tissues to lungs or alveoli. From tissues to lungs or alveoli. Now the third type, the third category of or third we can say rather category, third mode of transportation of carbon dioxide. We have already discussed that in soluble form. Now the third mode is in the form of carboaminohemoglobin, right? Put the third point. Transport of CO2. Transport of carbon dioxide as carboaminohemoglobin. Carboaminohemoglobin. Right. So what happened? Carbon dioxide carbon dioxide react with hemoglobin and form HbCO2, right? That is called carboaminohemoglobin. Every 100 ml of blood, every 100 ml of blood, 100 ml of blood deliver approximately 4 ml of CO2, 4 ml of CO2 to alveoli. Every 100 ml of blood approximately deliver 4 ml of CO2 to alveoli. Clear? Now let me make you more clear that what exactly we have studied. Right. In the part of the transportation of carbon dioxide, please try to understand. And in case if you have the this is the tissue, the respiration process is going on inside. We have studied respiration, glycolysis, and all things in detail. The same thing happened with the animal. Then the carbon dioxide comes out of the tissue. Carbon dioxide is transported to in the with the help of three modes: bicarbonate ion, HCO3 negative, and in solution form, solution carbon dioxide, soluble carbon dioxide, and again HbCO2 form. Right. Now all these mode of these are the just mode of transportation of carbon dioxide. So carbon dioxide is transported in these three forms: 7%, and that is 70%, and 23%. So all these form modes goes to, they have the common point of delivery, and that is the common point of delivery is your lungs. So they are going to deliver this carbon dioxide to your lungs. Now the lungs are the place where this carbon dioxide will be delivered. Now we have to study how they deliver this carbon dioxide to lungs, right? They have taken the carbon dioxide from tissue, how they are going to deliver to the lungs, right? So how they release the carbon dioxide inside the lung. So next heading will be release of CO2 in alveoli. I will put the next topic, release of CO2 in alveoli. That is the last topic, in fact. Release of CO, release of CO2. Right. Now, again, first I would like to explain the same thing with the help of that, uh, schematic. So this was my RBC. What happened in oxygen will enter inside the RBC. In RBC, now what will happen? So this RBC is released because of the entry of oxygen. Please try to understand. Suppose that when oxygen enter inside the, so when RBC goes to the lungs, what happened? When RBC will go to the lungs, what will happen? Then it will take oxygen, and when it will take oxygen, the bond of carbon dioxide become weaker, and it oxygen enter and it push out the carbon dioxide. That's what happened, and that's how the transfer of carbon dioxide takes place. So oxygen, let me write it down, from lungs. Oxygen from lungs or alveoli. Right? So oxygen will enter inside, and what will happen? There is HHB. HHB. We have studied HHB. Yes. So there is HHB. Now what will happen? This HHB combination will be broken down. Right. So the combination will broken down. Hemoglobin will become free, and hydrogen ion will be released. Now what will happen? This hemoglobin will make a bond with oxygen, and it will make HBO. So hemoglobin is engaged with now oxygen. Again, when these hydrogen ion will be released, right? This is the point what these hydrogen ion are released. These hydrogen ion, when the hydrogen ion will be released, then again the pump will become opposite. So I have already told you that there are certain antipode, right? They are exchanging something and taking in something. Now what will happen again? The hydrogen bicarbonate ion will start coming inside. HCO3 negative ion will start coming inside, which have been pushed out during the transportation. Now the chloride ion will go back to the plasma. Now the port, these port will become this will become it will start moving in opposite direction. Now this hydrogen ion, what will happen? Now this hydrogen ion, it will come to the bicarbonate ion, and these bicarbonate ion and hydrogen ion, they will combine again, and again they will make H2CO, means carbonic acid and unstable compound. So H2CO3 formation will take place. Again, I have told you that H2CO3 is an unstable compound. This complete reaction is taking place inside. Now what will happen? This H2CO3 will again break down in the presence of carbonic anhydrase. So the enzyme carbonic anhydrase, in presence of carbonic anhydrase, H2CO3 will break down into H2O plus CO2. Now the CO2 will be, now the CO2 is free. The CO2 will be, CO2 will be released in alveoli of lungs. That's how these RBC, they deliver carbon dioxide to the alveoli of lungs. Is it clear or not? Is it clear to all of you?
Yes, sir.
Yes,
Fine.
sir.
Very good, very good. So please note it down. And what we call it, actually, this is called Haldane effect. Mind it. This release of that was the hamburger effect, hamburger phenomena. This is called Haldane effect. So this process is also called Haldane effect. It is known as the Haldane effect because J.S. Haldane, a Scottish scientist, given the concept. J.S. Haldane. Please note it down, then I'll explain.
So this process will take place inside alveoli, right?
Exactly. No. Inside RBC. This complete process takes place inside RBC.
No, I mean like in alveoli or in tissues.
Sorry. No. This when this RBC will reach to alveoli, then RBC will take oxygen, and all the process then it will take place inside RBC.
Yeah, okay, sir.
Right. Done.
Yes, sir. Done. Yes.
Okay. So please start noting now below the diagram, below the schematic diagram. Binding of O2 with hemoglobin, binding of oxygen with hemoglobin tends to displace CO2. That is the summary of complete topic. Binding of oxygen with hemoglobin tends to displace CO2 from RBC. This is called Haldane effect. This is called Haldane effect. Let's stop. It is named after it is named after Scottish scientist G.S. Haldane. It is named after Scottish scientist G.S. Haldane. Next point. When hemoglobin in the lungs, when hemoglobin, when hemoglobin in the lungs, when hemoglobin in the lungs, blood capillaries takes of O2, when hemoglobin in the lungs, blood capillary takes of O2, comma, hydrogen ion are released from it. Hydrogen ions released from it. Full stop. Now, the chloride ion start moving out. Now, the chloride ion start moving out chloride ion start moving out from the RBC. Chloride ion starts moving out from the RBC, and bicarbonate ion and bicarbonate ion start coming in, and bicarbonate ion starts coming in RBC. In bracket, you can note it down. Opposite phenomena to chloride shift. Opposite phenomena to chloride shift. Next paragraph. After this, bicarbonate ion, these bicarbonate ion. After this, bicarbonate ion react with, after this, bicarbonate ion react with hydrogen ion to form H2CO3, carbonic acid, to form carbonic acid. Full stop. As a result, carbonic acid is split into H2O and CO2. As a result, carbonic acid is split into H2O and CO2 in presence of carbonic anhydrase enzyme. In presence of carbonic anhydrase enzyme and CO2. It is released to alveoli of lungs. It
Sir,
is,
can you repeat?
yes. As a result, H2CO3 is split into CO2 and H2O. As a result, H2CO3 is split into CO2 and H2O in presence of carbonic anhydrase, and CO2 is released into alveoli of lungs. Right? Now let's discuss about some of the defect or disorders. So till here, is there any problem? Both of the phenomena are crystal clear.
Yes.
Haldane effect is for Haldane effect have been asked in medical examination. Chloride effect many times it has been asked. Haldane effect have been asked rarely, but it is asked. And chloride shift is important for your school examination. Whenever I say that it is important for school examination, you have to tick over there, right? So you have to prepare that topic for your descriptive answer as well, right? Now put the heading respiratory disorder. I'm going to discuss some of the respiratory disorder with you. Then we'll go in next topic and so next heading is disorders. So out of this disorder, first I will discuss you some term. They are actually disorder. not only terms. So the first one is, what is hypoxia? What is hypoxia? Any idea? Note it down. It is the condition of it is the condition of oxygen shortage. It is the condition of oxygen shortage in tissue. It is the condition of oxygen shortage in tissues. Next term is that, what is anoxia? Note it down, guys. It is the absence of oxygen. It is the absence of oxygen in inspired gases. In inspired gases, comma, arterial blood or tissue. Inspired gases, arterial blood or tissues. That is anoxia. Now the third is mainly
Can you
phenomenon.
please repeat the last word?
Yes. It is the
Inspired gases.
absence of oxygen in inspired gases, comma, means either in inspired gases, arterial blood, means blood in auricle, or tissues. Next is proper disorder. So the third point is asthma. What is asthma? Very common disease, right? So please note it down. Allergent. So asthma takes place due to allergent. Allergent stimulate the release of histamine. Allergent stimulate the release of histamine. So what is the name of those cells which are found below the skin or at below the epithelial part, which release the histamine?
Can you repeat again?
What is the name of those
No,
cells?
like the sentence. First time.
Allergent stimulate the release of histamine. Allergent stimulate the release of histamine. Now my question was that which cells in our body release the histamine? Memuna. Anyone else? Look. Suppose that I have allergy with certain soap or certain shampoo or certain kind of the chemical, right? Suppose that I have a I want to color my hair brown, red, whatever I want. So one fine day, I tried that color. I brought a bright golden color, and I want to like color my hair. What will happen? Suppose that is allergen for me. I have allergy. When as soon as I will put the hair color, what will happen when it will come in contact with my skin? There are certain cells below my skin. They keep on wandering in that part below the skin, and the name of these cells called histamine. Sorry, mast cell. Name of these cells are mast cells. These mast cells will start releasing a substance called histamine, and after some time I will feel some itching like this way, and this complete part which is in contact with the that hair color, that will become red, and I will feel itching over there. That is a symptom of allergy, right? So allergy is created by mast cell, and they create the allergy with whenever we feel that itching taking place somewhere because of we got this some allergen, right? So these histamine cell, these mast cell, that releases histamine substance, and we get itching and all things because of the histamine. uh, Then what we do, we go to the medical shop or we go to the doctor and they give us anti-allergen. What anti-allergen do? They do nothing. They just calm down the mast cell to release the histamine, and release of histamine stops and we get relaxed. Clear? It is important. Separately, you have to remember this. Release of histamine from mast cell, release of histamine in mast cell causes inflammation in bronchi, causes inflammation, causes inflammation in bronchi, causes inflammation in bronchi, comma, bronchiole, bronchi or bronchiole. Full stop. Symptom: coughing, sneezing, and excess amount of coughing, sneezing, and excess amount of mucus clog, excess amount of mucus clog inside the lungs, right? Then this is important for both school as well as your medical exam, right? Debug disorders. The next disorder is, next disorders are occupational respiratory disorder. They are very important. They have been asked multiple times. Multiple times they have been asked. Occupational respiratory disorder. Occupational respiratory disorder. Actually, occupational respiratory disorder are the side effect of occupation, right? So suppose that you must have seen, whenever you go a furniture market and you want to bring a center table for your dinner table, right? Center table, dinner table. So they have a thick glass sheet, and that seems very beautiful. And that those people who are making the rubbing the glass, making the glass, they get infected with whenever they rub the glass and they want to make this edges smooth. What happens? The dust of the glass, it goes inside their nose and it causes them silicosis. People who are working with cement factory, silica keep on silica and asbestos keep on floating over here in the air, and lots of amount of the silica and asbestos goes inside their nose. They get infected with asbestosis or silicosis, right? Those people who are working in the cotton industry, right, they also get infected with silicosis. So these are some respiratory disorders, which, and these are the side effect of these occupation, right? So please note it down. They occur due to chronic exposure. They occur due to chronic exposure of. They occur due to chronic chronic exposure. They occur due to chronic exposure of harmful substances, harmful substances like harmful substances like silica, comma, asbestos, etc. etc. Next, proliferation of, now what happened? What is the symptom? Proliferation of means development of, proliferation of fibrous connective tissue, proliferation of fibrous connective tissue causes proliferation of fibrous connective tissue causes inflammation, inflammation in the lungs, right? Example,
What types of tissues? Connective tissues.
connective tissue, fibrous connective tissue causes inflammation in the lungs. Example. So what are the example of disease? Silicosis, asbestosis, asbestosis, etc. Next disease is pneumonia. Next disease is pneumonia. Pneumonia, that is a bacterial infection, you know. Pneumonia is caused by bacteria. Pneumonia is caused by a bacteria called Streptococcus pneumoniae. Pneumonia is caused by a bacteria Streptococcus pneumoniae, comma, other pathogen for it are fungus. Other pathogen for this disease are fungus. Alveoli inflated means the tightness or turgidity of alveoli decreases. Alveoli inflated, alveoli inflamed, alveoli inflamed, and most of space is filled with fluid, and most of space is filled with fluid and dead WBC. Full stop. Next is emphysema. Next is emphysema. It is caused exclusively by cigarette smoking. Cigarette smoking and like hookah and all that. Cigarette smoking and inhalation of other smoke. Cigarette smoking, and inhalation of other smoke or toxic substances. Cigarette smoking and inhalation of other smoke or toxic substances over a period damage alveolar wall. Full stop. Collapse of alveolar septa. Collapse
Repeat
of
from toxic substances.
toxic substances over a period damage alveolar wall. For sake of example, suppose that this is alveoli, right? This is a alveoli. Now the alveoli is like this. There is the septa, which keep it in shape. Then these are the septa when these septa will die or get collapsed, now the alveoli will, like, it will become flaccid, right? Substances over a period damage alveolar wall. Let's stop. Next point. Collapse of alveolar septa reduces the surface area reduces the surface area of alveoli reduces the surface area of alveoli for the gaseous exchange. Full stop. These were some respiratory disorders. So, guys, go through with it and let me know in case if you have any doubt, any question. And
Sir, can you please repeat the last sentence?
which one?
Collapse of alveolar septa.
Collapse of alveolar septa reduces the surface area of alveoli for exchange of gases. Clear?
Yes, sir.
Now, let me know if you have any question, any doubt, then we will proceed further in next topic and next.
Sir, can you explain the anoxia?
Anoxia is just, look, this anoxia is absence of O2, means O2 is not present. We are inhaling the gas, but O2 is not present. Whenever we inhale the gas, 21% O2 is present over there. Then O2 is not present over there. Rest of the gases are present, right? First condition. Second condition, we have breathed in O2, but oxygen is not present in the atria, means auricle of our heart. Third, oxygen is not present, right? O2 is not present in the tissues, right? So in any part of the body tissue, if O2 is not present, so absence of O2 either in the gas we are inhaling, or in the tissue, or in the atrial blood. All these conditions, all these three conditions are called anoxia. Clear?
Yes, sir.
Anyone else? Any more question? No question? So before moving, the next chapter is, I think it's body fluid and circulation. Yes? Is it that? Yes? That is the chapter? Guys, please look at the NCERT. Yes,
Yes, sir.
Pramana? Yes.
Yes, sir.
Okay, fine. So before going into that chapter, I want to see in front of you, I want to see the assignment which you did in last class. I'll open the assignment in front of you, which you people have submitted, who are there. The students assignment. So, okay, the assignment of Mamuna is open. Mamuna,
Yes, sir.
is that the last assignment?
I think no. We did after that also.
Which one?
There were, I think, about 20 MCQs.
MCQ, written question. Somewhere this is the locomotion. Okay. Digestive system. What was the last assignment that you did, which you submitted actually to discuss it?
Yes, so this was the last one.
Digestive
Yes.
system. This was the last one.
Yeah.
Okay, fine. So, okay, so this is not a descriptive one. Fine. Let me open the
The transport of gases was the last one.
transport of the gases. When you given it? When you submitted it actually?
1 minute, sir. Sir, I guess 13th.
13. Okay, 13. Oh, so that was, and the assignment was respiration, from respiration only. Transport of gases. Okay, I'll ask him to upload it. We'll discuss on Wednesday. No issue, because I have the assignment in front of me. That is this one, and this one, from the digestive system, and he have to upload with the. Let me check it. Is it there? No. This belongs to digestion. And keep on writing down something like you given this assignment, this one, right? This assignment, right? This is the way of writing, and this is the assignment actually, where we get the chances to evaluate. Else in MCQ, what I can do, just I can tick either it's wrong or it's right. But in descriptive question, I came to know that how good you have hold in your conceptual clarity, right? So I'll ask him to upload that, and we'll discuss in Wednesday as well, because this is not right now here. So the next topic which we are going to discuss, or which we are going to start, that is the blood circulatory system. Fine. So, this is your batch. So next we are going to do circulatory system, body fluid and circulation actually. Body fluid and circulation. So in body fluid and circulation, I'll tell you something, some factual thing. What we are going to study actually, this is our circulatory system, right? So first, let's study the composition of blood, right? Human blood and its composition. Not human blood, human fluid. Human fluid and composition. Right. We are going to discuss this thing first, and then I'll start here. I'll tell you some factual thing which is important for medical in this chapter, right? And then I will tell you what is the circulatory system, how the fluid. So before going deep into that thing, that we have two kind of the fluid, right? ECF and ICF. What is ECF and ICF, right? So if we talk about the human fluid, we are just talking about the fluid which is found in our blood. Human body fluid. So out of there are two kind of the fluid, ECF and ICF. ICF, right? ECF is extracellular fluid. Extracellular. What is the extracellular fluid? Extracellular fluid is the lymph. That is the blood. So blood and lymph are two things which are extra. They are found out of the cell. Extracellular fluid means they are found out of the cell, outside of cell. And ICF, that is intracellular fluid. Intracellular fluid. In intracellular fluid, there are just protoplasm or cell sap is not found in our body. So this is protoplasm. So that is the ECF and ICF. Now we are going to study the ECF. So in ECF, we will study the lymph and the blood. What is the difference between the lymph and blood? What is the composition of lymph and blood? We'll discuss all things in detail. Then we will discuss the circulation of these fluid, like how does these fluid circulate in our body, how does they deliver the substances, what kind of the substances they deliver, and all that, right? So before going deep into the topic, please note down some factual things in the form of pointer, right? So the first point is that, tell me, the question have been already asked. What percentage of water is found in our body? Tell me.
60%.
Absolutely right. 60% of body
Of water.
means 60% of mass, weight, volume. You are absolutely correct. 60% is the answer. Yes, Mamuna. What should be? Volume, mass, or weight? Weight. Right? So please note it down. Water is about 60% of body weight. Water is about 60% of body weight, or 42 liter in human, or 42 liter in a human of average weight, in a human of average weight 70 kg.
Sir, can you repeat from 60% of body weight?
Water is about 60% of body weight, or 42 liter, or 42 liter in a human of average, 70 kg weight. Next point. An increase in the percentage of fat, an increase in the percentage of fat leads to decrease, an increase in the percentage of fat leads to decrease the amount of water, leads to decrease the amount of water. Next point. Intracellular fluid means the fat people will have less water in their body. Non-fatty people will have more water in their body. Intracellular fluid, ICF means. Intracellular fluid contains large amount of large amount of potassium and phosphate, large amount of potassium and phosphate ion, large amount of potassium and phosphate ion. Full stop. Next point. Sodium and chloride. Sodium and chloride ions are important constituent. Sodium and chloride ions are important constituent of ECF. Next, very important point, make a star. Make a star. Blood. Yes. Make a star. Blood forms 30 to 35%. Blood form 30 to 35% part of ECF. Blood form 30 to 35% part of ECF. Full stop. Next point. An adult person of 70 kg weight, an adult person of 70 kg weight contain approximately 5.5 liter of blood, 5.5 liter of blood with pH value 7.4, 5.5 liter of blood with pH value 7.4. Next. Now come to the plasma. This question I have asked in like one of the exam. Now plasma. Plasma means blood plasma, right? Plasma is alkaline. Plasma is alkaline, non-living constituent, non-living constituent, which makes non-living constituent. Please underline non-living. This was the question actually. Non-living constituent, which makes 55% part of blood, which makes 55% part of blood. Right? So that question have also been asked 55%. Underline the 55% and non-living. These are the two questions which have been asked in medical examination. Next, water forms. So plasma is 55% of blood. Next, the water alone forms 92%. Water alone form 92% part of plasma. Part of plasma. Water alone forms 92% part of plasma. Next. Plasma contain mineral. Plasma contain mineral, nutrient, plus protein. Plasma contain mineral, nutrient plus protein. Next point. It contain which plasma? It contain 6 to 8%. It contain 6 to 8% protein of plasma. It contain 6 to 8% protein of plasma, which is albumin, and globin, Now I have a question, and this is very important, very good question. The pH value of blood, as we have discussed number of the time, like here, even in respiration part, I have discussed that 7.4. Here I am discussing 7.4. So pH value of blood is 7.4. That is the basic. Now, whatever you eat from the morning to the evening, that is all acidic. You take water, water is not actually neutral. The water we take, that is mineral water, that is acidic in nature. You eat carbohydrate, means sugar, that is acidic. You eat fat, fatty acid, that is acidic. You eat protein, that is amino acid, that is acidic. So everything which is tasty, which appears tasty to us because acids are tasty, but the bases are very sour and bitter kind of the taste. They have very bad taste. So everything, whatever we eat from morning to evening, that is acidic, that get absorbed in our blood, but still the pH value of blood do not decreases. It always remains basic. So in spite of eating so many things, everything in acidic form, why our blood is basic? And which is the thing which maintain the basicity or pH value of the blood? That is a question. Name the thing which maintain the pH value of blood. Who is going to tell me? Come on. Tell me one very simple question. What is albumin protein? Do you have any example of albumin protein? Come on, guys. An example of albumin protein. The white part of egg is made up of albumin. Yes, white part of egg is made up of albumin. Actually, albumin protein work as buffer solutions. Buffer solutions are those which do not allow to change the pH value of their solutions, and they always maintain the pH value. Albumin protein is found in the blood plasma. It do not allow blood to change its pH value in spite we eat everything which is acidic in nature. Clear or not? Did you got my point?
Yes, sir.
Make a star below here and please note it down right. Albumin, in bracket, please note down egg white protein. It is found in egg white. Albumin is found in blood. It works as buffer solution found in blood. It works as buffer solution and it do not allow blood to change its pH value. It do not allow blood to change its pH value. It do not allow blood to change its pH value. Next point. Lysozyme or properdin. Let me write it down. Lysozyme, or there is another thing, and not or, lysozyme and properdin. Properdin. Lysozyme and properdin. Please note it down. Lysozyme and properdin are also found in blood plasma. These are two enzymes. Lysozyme and properdin are also found in blood plasma, which destroy bacteria, virus, and toxic substances. Lysozyme and properdin are also found in blood plasma, which destroy bacteria, virus, and toxic substances. Full stop. What is heparin? Can you tell me? Anybody? What is heparin? Heparin is a type of hormone. What is the function? Like why it's important? Heparin is a hormone, not enzyme. Found in our blood. What is the relevant of? Look, heparin do not allow blood to clot inside our body. That's why it is called anticoagulant. Frequently asked question. It is called anticoagulant. Just a minute. This is called anti. It is called anticoagulant. Or it is called anti-clotting agent. Very frequently asked question. Please note it down. Heparin is a hormone found in our blood at dissolved state. Heparin is a hormone found in our blood at dissolved state. It do not allow blood to clot inside our body. It do not allow blood to clot inside our body. That's why this is called anti-clotting agent. That's why this is called anti-clotting agent or anticoagulant.
So can you repeat?
Yes. Heparin is a hormone found in our blood.
So from that's why, sir. Like
That's why it is called anti-clotting agent. I have written it down. That's why it is called anti-clotting agent or anticoagulant. What is blood serum? You must have listened about the blood serum. When you remove this heparin from the blood plasma, that is called serum. So note it down. The blood plasma without clotting factor. The blood plasma without clotting factor is called blood serum. Blood plasma without clotting factor is called blood serum. Right? So these were some
sir, as you have said that heparin it prevents the internal clotting, but if sometimes there is like an internal bleeding, sir, the blood clots inside, like inside the nail, sir.
yes, I will tell you like why the clotting takes place and how does chemically actually clotting takes place. I will tell you a separate topic for that, and that is very important for your school examination as well, right? And that is very important for your medical examination. Clotting, how the clotting takes place, right? What are the events which takes place in clotting? I'll discuss that, right?
Okay.
Actually, in short, I want to tell you, whenever a molecular oxygen enters in body, whenever we get like a rupturing takes place, right? Whenever the tissue get ruptured, so what happened, that molecular oxygen enters over there, and due to entry of the molecular oxygen, heparin stops working, and when heparin stops working, clotting takes place. That's how the clotting takes place. So that was in short, but I'll tell you in detail the clotting process. I'll explain you in detail topic, right? And again, in short, I want to tell you what is actually clotting or coagulation process. How does the clotting or coagulation process takes place? So, in short, or rather than the clotting, I want to tell you what are the component of the blood. For sake of example, we have glass. and that glass is filled with the blood. This is a tumbler, and this tumbler is filled with blood, right? So for example, this is a tumbler, and this tumbler is filled with blood. This is blood in tumbler, right? If this blood is of a healthy person, right? So this was a blood and filled in a tumbler, and this, if this blood, please listen whatever I am telling you carefully. If this blood belong to a healthy person means who do not have any disease, right? After the 7 to 20 minutes, the average time is 6 to 7 minutes, 6 to 7 minutes, and maximum time is 20 minutes maximum, and this is average time, right? So if this blood belongs to a healthy person, what will happen after some time? After some time means after say 6 to 7 minutes, the changes will take place over here. Now what will happen? This red part will become a ball-like structure that is called clot, right? This is a ball-like structure, right? This is a ball-like structure. This is a ball-like structure, and the rest part will be the rest part will get separated from it. So the rest part is get separated. Rest part is getting separated from it. So rest part will be a slightly yellow color substance, not that much yellow, which is visible at your skin, not that much, slightly yellow colored liquid. So this slightly yellow colored liquid is actually called blood plasma. So blood plasma gets separated from the clotting part or the solid part. So this process is called actually blood clotting or blood coagulation. That's how the blood clotting or blood coagulation takes place, right? So entry of molecular oxygen. So that is molecular oxygen. Molecular oxygen will enter and clotting will take place. This part is actually called blood clot. So whenever we get injury, what happen? Molecular oxygen enter over there, heparin stops working, and blood clot will take place. Now, what are the component of this blood clot? My motto is not over here to tell you the blood clotting, because blood clotting I will discuss you in detail. But here I want to tell you that what is the percent. The question which have been asked in medical examination. This is blood plasma. So the question was that what is the percentage of plasma in blood? I have explained you right now. I have explained already. What is the percentage? Hey, come on. You have noted down the point right now.
90 to 90.
90 to 90.
90 to 92%.
Oh, come on. Read it again. That is a percentage of water in
Oh,
plasma.
sorry, 55%.
Yes, 55%. Right. So the blood plasma is the 55% of the blood. The rest, the clotting part contribute 45%. Now, what is the composition of this 55%? Out of this 55%, 90 to 92% is water. Then what is the rest component? Rest component is carbohydrate, means sugar, then protein, then fat, then mineral, right? Mineral and then hormone. They are the component. They are the component of actually plasma. What is the factor of blood clot? There is RBC, WBC, platelet, then some fibrous protein, or then thrombin protein, then fibrin protein. These are the main component of, please jot it down this one. These are the main component of blood. These are the part of actually blood. Now I think it must be clear to you that what is the actually blood plasma is. Then we'll start studying from the RBC onward.
So what's it after platelets?
Sorry, after protein, there is a thrombin protein, thrombin and fibrin or fibrinogen protein.
So what's the first
First one,
in blood plasma?
RBC, WBC, platelet, thrombin protein, and fibrin protein. And above, this is the hormone, carbohydrate, protein, fat, and mineral. Done?
Yes.
Okay. Should I move into next slide? Miss, next page?
Yes.
Fine.
Yes, sir.
Okay, great, great. So the next thing that we'll study RBC, WBC, and platelet, right? Then we'll go to into next topic. Right? So first, the next heading is RBC. Put a next heading. That is RBC. That was human fluid, and I must write it blood composition. So next heading will be RBC. So RBC is red blood corpuscles. Red blood corpuscles. Corpuscles. So my first question is that why we call them red blood corpuscles? CARPUS corpuscles. Why do not we call them CARPUSCLAs? Okay. Yes. Why we call them corpuscles? Why not cell? Red blood cell. If red blood cell is written over anywhere, then that is wrong. Let me tell you, that is completely wrong. RBC is not red blood cell. Look, to become cell, there are certain conditions. It should have nucleus, it should have some cell organelle, it should have all cell organelle, it should have all those components, endoplasmic reticulum, Golgi complex, and all that. RBC, WBC, like these blood cells do not have all these components. That's why we cannot give them the designation of cell. So most of the cell organelle remains absent in corpuscles. That's why we use term corpuscles. We cannot use the term cells over here. Blood cells. This is the actually RBC. This is actually a diagram of red blood corpuscles, RBC. And RBC have some hemoglobin molecule. It is also known as erythrocyte. So RBC, let me make you clear that RBC is the another name of the RBC is also called erythrocyte. Erythrocyte. Right. So there are some factual things. So they are disc-shaped structure and they are biconcave. Means you must have seen the Alpen toffee. They look like Alpen toffee. They are both of the surfaces are concave and they are disc-shaped structure and they have hemoglobin. Hemoglobin is made up of two things. First, I'll explain you. Then hemoglobin is made up of two things: a protein component and heme part. Right. So actually, hemoglobin is made up of globin protein. Globin is globular protein and iron component, and collectively both are called hemoglobin. Right here, the oxygen combine with hemoglobin molecule and form the oxyhemoglobin. That's how they carry oxygen. We have discussed in previous chapter, so in detail. So please start noting down. A normal man or woman have a normal man or woman have 4.5 to 5 million 4.5 to 5 million RBC per mm cubic. A normal man and woman have 4.5 to 5 million RBC to normal man. Next, important point is that abnormal rise in RBC, abnormal, so that is a disease. There is also disease, rise in RBC. Abnormal rise in RBC is called polycythemia. Is called polycythemia. This disease is termed as polycythemia. Polycythemia means abnormal increase in RBC. Abnormal rise in RBC is called polycythemia, and abnormal decrease in number, abnormal decrease in number is called erythrocytopenia. Erythrocytopenia. And abnormal decrease in RBC is called erythrocytopenia.
Sir, like what happens if the RBC increase, sir?
Nothing. I just most of the and then the carrying capacity of oxygen decreases, right? So normally one RBC can carry a particular amount of the oxygen, right? They have a 4 hemoglobin molecule. One hemoglobin molecule can carry approximately 4 oxygen molecule, right? So when the number of RBC will increase, then every hemoglobin molecule will carry 2 oxygen molecules. So that is a polycythemia. Polycythemia is not that much fatal or that much like uh, harmful for our body, but erythrocytopenia is really harmful for body, right? Next one, very important point. This is common point, but I have been asked number of times. Non-nucleated RBC are non-nucleated. They do not have nucleus. Mature human RBC do not have nucleus. They are non-nucleated. That is, do not have nucleus. Biconcave in shape. Non-nucleated. That is, do not have nucleus, and biconcave in shape. Biconcave in shape. Size varies from 7 to 8 micrometer. Size varies from 7 to 8 micrometer in diameter. Mature mammalian RBC, mature mammalian RBC do not have cell organelle. Mature mammalian RBC do not have cell organelle, including nucleus. Full stop. 100 ml of blood, 100 ml of blood of a normal individual, 100 ml of blood of a normal individual contain about 20 to 16 gram, sorry, 12 to 16 gram, 12 to 16 gram of hemoglobin. 100 ml blood of normal individual contain about 12 to 16 gram of hemoglobin. So when we remains inside our mother's womb, then on that condition, where RBC are produced in our body? Very good question. Make a star and tell me. Then you have to note it out. So in embryonic stage, where the blood is produced? Where the RBC is produced at adult stage or mature stage? Come on, guys, don't tell me you don't know the answer of this question. Yes, Mamuna, where the RBC is found in our body? Come on, guys. Yushra? In our bone marrow. RBC is found in our bone marrow. But during the embryonic stage, RBC is not found in the bone marrow. So what we call the formation of blood or formation of RBC, that is called erythrocytopenia, right? So formation of erythropoiesis. So formation of RBC, the process is called erythropoiesis. Erythropoiesis. This is the formation of RBC. Please note it down. The formation of RBC is called erythropoiesis. Formation of RBC is called erythropoiesis. In adult stage, it takes place at bone marrow, full stop. But at underline this statement, at embryonic stage, RBC are produced in our liver. At embryonic stage, RBC is formed in our liver. Full stop. The lifespan of RBC is 120 days. Lifespan of RBC is 120 days. Next, deficiency of RBC leads to anemia. So after 120 days, RBC dies. In a nutshell, every day approximately 80 lakh RBC dies on our body. What happened to these dead RBC? Where they goes? I have a straight question. Which organ is called graveyard of RBC? Any idea? Graveyard of RBC. We have an organ called spleen. Spleen is called graveyard of RBC. All the RBC after death goes to spleen. One more thing. Extra RBC are stored in their spleen as well, right? So this is called graveyard of RBC. This is also called blood bank. Spleen is called graveyard of RBC and blood bank. Clear? Note it down. Spleen is called graveyard of RBC because spleen is called graveyard of RBC because all the RBC reaches to spleen after death. All the RBC reaches to spleen after death. Full stop. The spleen is also called blood bank, Clear? is
Yes,
gas.
sir.
So that's it for the day today. And in the next day, like Wednesday, we'll finish, most probably we'll finish this chapter, right? And in case if you have any doubt, any question, please ask me. Please, come on. Any doubt? Any question?
No, sir.
No? So that's it for the day. See you on Wednesday, same time, and we'll continue with the topic. Fine?
Yes, sir.
Bye-bye. Take care. Good night.
Excuse me, sir.
Yes.
What is deficiency of RBC called?
Anemia. Anemia. That is called anemia. If it goes down 12 to 14 gram, 12 to 16 gram, so if it is below the 12 gram, that is anemia. Right? Clear? Any more question? So till the time, bye-bye. Take care. Good night. And see you in
Thank you,
next
sir.
class. Yes. Welcome. Bye-bye.
Thank you, sir.
Welcome, beta. Welcome. Bye. Bye.